Have you ever wondered what the world would look like after humans are gone? Well, scientists have been thinking about that too. Without us, the planet would likely look very different, and certain creatures could evolve into something far stranger than we could imagine. From aquatic beasts to land-dwelling giants, nature might take a few unexpected turns. Today we are going to dive into fascinating creatures that could evolve in our absence. These aren't just creatures from our imagination, they're based on real science and evolutionary theory. Let's get started. Imagine giant squirming squids roaming the earth. Sounds like a nightmare, right? But scientists say it could become a reality. Squids and octopuses are already some of the most intelligent creatures on Earth, but once humans vanish, their evolution could take a fascinating turn. Squids are already skilled in adapting to various environments, changing colors, textures, and even regrowing limbs. With no humans to disrupt ecosystems, squids might develop larger, more complex forms capable of thriving on land. These creatures could evolve into terrestrial predators using their impressive problem-solving skills and adaptability to dominate environments. We could see them with stronger limbs, possibly resembling the tentacle-like appendages of cephalopods that would allow them to move on land with an efficiency that is hard to imagine. And considering how many of today's largest animals evolved from smaller ancestors, it wouldn't be too far out of the realm of possibility to envision squids the size of modern-day elephants. So squids are just in it for the long game. You know, they're just biding their time, waiting for humans to disappear, and then they rule the land. What if the next creatures to inherit Earth's intellectual throne aren't apes, but a completely different species? With humans gone, it's possible that mammals, like dolphins, elephants, or even rats, could evolve to become hyper-intelligent. These creatures already show signs of sophisticated communication and problem-solving abilities. In a world without humans, they might develop complex societies and technology, possibly even communicating through advanced systems or tools. Imagine dolphins building cities underwater, or elephants mastering land navigation and technology. What if the birds we see today are just a glimpse into the future of avian evolution, where birds grow to dominate the skies once again? Birds are the direct descendants of dinosaurs, and after humans are gone, many experts believe that birds could evolve back into larger, more dinosaur-like creatures. In the absence of humans, birds might grow to unprecedented sizes, filling niches left open by the extinction of large terrestrial animals. These bird dinosaurs could resemble some of the giants from the Mesozoic era, such as the Argentavis, an ancient bird with a wingspan over 23 feet. What would happen if rats, the survivors of the apocalypse, grew in size? You might want to keep your distance. After humans are gone, scientists speculate that rats or similar rodents could evolve into much larger, more dangerous creatures. With a high reproductive rate, rodents would quickly take advantage of available resources, evolving into new forms with larger bodies to adapt to new environments. Imagine a world where giant rats or squirrels scurry about, competing for food and resources, dominating the landscape just as they did in the early days of mammals.
Passenger pigeons used to be everywhere in North America, millions of them flying in huge flocks. They'd roost in forests so thick that branches would break and drop tons of pigeon droppings, which is gross, but it actually helped fertilize the soil. By 1914, they were totally wiped out, thanks to overhunting and habitat loss. But now, scientists are trying to bring them back by using DNA from museum specimens and editing that into their closest living relative, the band-tailed pigeon. Because band-tailed DNA isn't identical, this would create a hybrid. Not a perfect passenger pigeon, but it would be about as close as you can get. They also tested how reintroducing these birds could affect forests. For example, researchers in Michigan simulated how a massive flock might disturb the trees, breaking branches, letting light in, changing the soil, in order to see if these birds would actually be helping anything. Right now, the team has band-tailed pigeons with CRISPR gene editing tools built in so that the edits can be passed to their offspring. And they're aiming to hatch the first passenger pigeon-like chicks around 2029, before eventually testing releases back into the wild. The Pyrenean Ibex, also called the bucardo, was a wild mountain goat that lived in the Pyrenees Mountains between Spain and France. It was extinct in 2000 when the last known female, named Celia, died after getting caught under a fallen tree. But scientists were not ready to give up on the species. In 2003, researchers used frozen skin samples from Celia to try cloning her. They took DNA from those samples, inserted it into goat eggs, and implanted the embryos into domestic goat surrogates. For a brief moment, it worked. The cloned ibex was born, the first time an extinct animal had been brought back to life. But the clone only survived about 7 minutes. It had really bad lung problems, so technically the Pyrenean ibex went extinct 2 times. But the experiment was not a total failure. It proved cloning was possible, it just had to be perfected. The northern white rhino was nearly extinct by 2018. There were only 2 left in the world, and they were both female, so it really looked like this was it. But scientists in Kenya, along with members of the Leibniz Institute for Zoo and Wildlife Research in Germany, and the San Diego Zoo's Frozen Zoo, collected eggs from the 2 surviving females and used frozen sperm from males that had already died. The plan is to make embryos in the lab and implant them into southern white rhino females who can act as surrogate moms. That way the babies would be northern white rhinos just born from a closely related species. The process is not easy though. Rhinos have really long pregnancies, around 16 months, and getting in vitro fertilization to work in rhinos is super tricky, but the team's already made some healthy embryos and now they're focused on getting one of them to actually develop into a living calf. If this works, the northern white rhino would go from being basically extinct to having a real chance at survival again. It's a long road, but it's probably the species' only chance. Next up we have Dolly the sheep. Now obviously sheep aren't extinct, but this was the first mammal cloned from an actual adult cell. This changed everything. Back in 1996, scientists took a cell from a Finn Dorset sheep and transferred its nucleus into an egg cell that had its own DNA removed. This egg was then implanted into a surrogate sheep, and out came Dolly, the world's first cloned mammal. She wasn't the first animal cloned overall, but cloning from an adult cell showed that mature cells could be sorta reprogrammed to develop into an entire animal. Dolly lived for 6 years, had 6 lambs. Cloning was now more than just science fiction. By the late 80s, black-footed ferrets were basically extinct in the wild. Luckily, a small group was found in Wyoming in 1981, and conservationists captured some to start breeding them in captivity. But since the population was so small, the ferrets' genes were too similar. Too much mixing of similar genes is obviously not a good thing. There needs to be some variation in there. So in 2020, the group Revive and Restore cloned a ferret named Elizabeth Ann to help fix this, using frozen cells from a wild ferret. Elizabeth Ann was born healthy and released back into the wild to add some new genetic variety into the ferret population. The Przewalski's horse is the last truly wild horse species on Earth. Wild meaning it was never domesticated. These horses used to roam the steppes of Central Asia, but by the late 60s, they were pretty much mostly wiped out. The main reasons were overhunting, plus fences and farms pushing them out of their habitats. After that, the only Przewalski's horses left were stuck in zoos and breeding programs. But in 2020, scientists used frozen tissue from a stallion named Kaporovic, stored for 40 years in a frozen zoo, to clone a healthy baby named Kurt in Texas. A few years later, they cloned Kurt's genetic twin, Ollie. These clones brought fresh genes into the very small population, and now these horses are being released back into the wild. This is probably the most successful de-extinction case in history, hopefully not the last. Glowfish are probably one of the most famous examples of genetically modified pets. These little fish, which are actually zebrafish, were engineered in 1999 to glow in the dark. The trick? Scientists inserted a jellyfish gene into the zebrafish, causing them to produce a fluorescent protein that makes them glow under ultraviolet light. The idea behind the glowing fish was actually pretty practical at first. The researchers wanted to use them as a way to detect water pollution. If the water was contaminated, the fish would change color. But as often happens with things that are cool and unusual, people started getting getting more interested in these things as potential pets. And by 2003, glowfish were available for sale in the United States, and they quickly became a novelty item. But their release raised some ethical questions. Some people thought it was just fun, but others were uncomfortable with the fact that these were pets that were cooked up in a lab. Plus, there were worries about what might happen if these fish were released into the wild, where they could potentially disrupt ecosystems. Glowfish are legal to sell in many places, but they are banned or restricted in others. At the Mayo Clinic in Minnesota, scientists injected human stem cells into pig fetuses. What they ended up creating was something that could be described as the first pigman, or a pig with human traits. The goal of the experiment was to study how human and pig cells would interact when fused together, because why not, I guess. What they discovered was pretty wild. Some cells kept their separation, so in the pig's body there were areas with just human cells and others with just pig cells. But other times the cells actually fused together and formed a brand new type of DNA that no one had ever seen before. So on the outside it was a regular pig, but on the inside it was something much more complicated. An animal with half human, half pig cells throughout its body. The most interesting part was the BLOOD, which was a hybrid of human and pig, and this was a completely new type of BLOOD that had never existed before. Scientists were able to confirm that this was the first time such an experiment had ever been done. Now this one's pretty mind-blowing. For a long time, scientists have been piecing together how dinosaurs evolved into modern birds. And in 2016, a team of researchers in Chile set out to watch this evolution in real time by experimenting with chicken embryos. Their goal was to look at how the leg bones of birds have changed over time and how they connect to their dinosaur ancestors. Dinosaurs, like the ones we see in fossils, had two long bones in their lower legs, the fibula and tibia, which ran parallel to one another. Over millions of years, those bones evolved. When it came to modern birds, the fibula became much shorter and developed a splinter-like end, while the tibia became the dominant bone in the leg. Now the researchers wanted to see if they could make chicken embryos grow that older dinosaur-like leg. So they took a gene that plays a role in this leg development, the IHH gene, and suppressed it in the embryos. And the chicken embryos developed leg bones that looked a lot more like dinosaur legs, with both bones being of similar length and reaching down to the ankle. These dino chickens didn't survive long enough to hatch, but this was the first time scientists could directly observe how bird legs evolved from dinosaur legs in living creatures. Before this experiment, it was just all speculation from fossils. Back in 2018, researchers produced sheep-human embryos. They wanted to see if they could grow organs inside these engineered animals. The end goal is to have body parts that could one day be used for organ donations. Scientists introduced human stem cells into sheep embryos, and while the embryos were mostly sheep, a small fraction, less than 1%, contained human cells. Stem cell biologist Hiro Nakauchi from Stanford University said, "The contribution of human cells so far is very small. It's nothing like a pig with a human face or human brain." And yeah, it was not that, fortunately. Yeah, not a huge percentage here, but the fact that they were able to create sheep embryos with even a small fraction of human DNA is pretty wild. The sheep embryos were destroyed after 28 days, so no sheep-human hybrids were actually born, but the experiment showed that creating organs inside animals with human DNA could be a possible solution to the growing need for organ donors. Oh, Florida, the state has enough wild stuff to worry about than, super snakes, but hybrid pythons are a real problem in Florida. These snakes are massive. Some could grow up to 23 feet long and weigh nearly 200 pounds. Originally, a lot of these snakes were exotic pets, but over time, a lot of them were released into the wild. And by the late 80s, the Burmese pythons had grown into a pretty sizable population in Florida and became a massive problem for the ecosystem. But even worse, researchers studying the pythons in 2018 found something pretty unsettling. Out of 426 snakes they sampled, 13 were hybrids. These hybrids were a mix of Burmese pythons and another species, the Indian python, which had also been released by pet owners. The two species hadn't just recently mixed, they'd been interbreeding for a long time before researchers discovered them. The Indian python is smaller but quicker than the Burmese python, and this combination makes them tougher and more adaptable.
Coming in at number 5, Aurochs. Considered to be the wild ancestor of modern domestic cattle, Aurochs were a species of large cattle that once lived in Europe, Asia, and North Africa. And when I say large, I mean large. Apparently Aurochs could reach a height of 6.6 feet at the shoulder and weigh up to 3,000 pounds. However, due to hunting and habitation loss, the Aurochs became extinct when the last individual died in 1627. However, recreating the extinct species may not be an out-of-this-world thought according to scientists. Starting back in the 1920s, Heinz Heck, a German biologist, initiated a selective breeding program where he attempted to breed back the Aurochs using several cattle breeds. Essentially, the idea was to breed out what they had been domesticated for, and the result was called Heck cattle. By the 1980s, herds of Heck cattle were released into the Netherlands, and since, Heck cattle have been crossbred with other European cattle breeds in the hopes of creating a more Aurochs-like cow. Coming in at number 4, the dodo. Maybe the most recognizable of any extinct species due to just how straight up bananas this bird looked was the dodo. Once upon a time it was found exclusively on the island of Mauritius, but what was most fascinating about the dodo was that it evolved without any natural predators. However, like many animals that evolved in isolation, the dodo was entirely fearless of humans. However, this fearlessness, coupled with its inability to fly, unfortunately made the dodo an all too easy target for sailors. Although contrary to popular belief, it wasn't just hunting that took out the dodos, but the introduction of other animals like monkeys and pigs into their habitat, who destroyed their nests and forced them into a competition for limited resources as well. So, have these cartoon-looking birds actually been brought back since their 17th century extinction? To be honest, not technically. But scientists are trying. Beth Shapiro, a professor of ecology and evolutionary biology at the University of California, said that she has completed fully sequencing the dodo's genome from an ancient DNA based on genetic material extracted from dodo remains, and the next steps are in the works. Next up at number 3, the Tasmanian tiger. The Tasmanian tiger was once a carnivorous marsupial native to the Australian mainland as well as the islands of Tasmania and New Guinea. But beginning in the 19th century, they were perceived as a threat to the livestock of farmers, so overhunting was eventually the largest factor in their demise. Sadly, the last of the Tasmanian tigers was illegally captured in 1936 by Elias Churchill and sold off to a zoo where she stayed until her death. But a return of the Tasmanian tiger could very well happen in our lifetime. In 1999, the Australian Museum began a cloning project for the animal, an endeavor that was initially dismissed as a publicity stunt. But in 2002, researchers successfully extracted replicable DNA from the specimens. Now fast forward to August of last year, and the University of Melbourne announced it will be partnering with a company called Colossal Biosciences to attempt to recreate the animal using its closest living relative and return it to Tasmania. Coming in at number 2, the woolly mammoth. I'm sure we're all familiar with the gigantic fuzzy ancestor to the elephant because, well, it's probably one of the most famous extinct species recorded. The last isolated population of woolly mammoths lived on Wrangel Island in the Arctic Ocean until roughly 4,000 years ago, and famously the first actual remains of the behemoth were found in Siberia in the late 18th century. Interestingly, at first, the researchers studying them were incredibly confused because they could not understand how a warm climate animal such as an elephant could have wound up in such a freezing cold area. But eventually they put all the pieces together and came to the realization that what they had found were the remains of an extinct animal. Amazingly, since those first few skeptical years, thanks to the cold temperatures in the Arctic, the carcasses of the extinct creature have been super well preserved by ice, allowing scientists to access the DNA in ways that is not always possible. And thanks to that, it is apparently in the works to try and bring the beasts back into the world. In fact, after a genome project for the mammoth was completed in 2015, it has been proposed the species could be revived through various means. So while it hasn't happened just yet, by the sounds of it, it could be any day now. And last up in our number 1 spot, the Pyrenean ibex. While there are many genetic testings in the works along with breeding of similarly extinct species with the hopes of adaptation, if we are talking about an actually extinct animal that has been successfully brought back to life, then the Pyrenean ibex is in a league of its own. During its prime, it was often found in areas like France, Portugal, Spain, and Andorra. However, starting in the late 19th century, their population began to dwindle until eventually the Pyrenean ibex was officially declared extinct in 2000. But the amazing thing about this animal is that unlike any of the others on this list, 3 years after the last one died, scientists used its frozen cells to clone a calf, making it the first and only animal to have a living specimen exist post-extinction. Now, the caveat is that sadly the clone did not survive long after birth. It actually died a few minutes later due to a lung defect. But it did prove that it could be possible to actually bring a species back from the dead.
Colossal Biosciences, a Dallas biotech founded in 2021 by George Church and Ben Lamb, actually brought the dire wolf back from extinction. They took ancient DNA, one sample about 13,000 years old, the other around 72,000, identified about 20 genes that gave the ancient wolves their larger skulls, stronger bones, lighter fur, more muscular legs, and then they used CRISPR to edit those into modern gray wolf cells. They cloned those edited cells via what's called somatic cell nuclear transfer, transferring the edited cells into dog embryos, which were carried by domestic dogs. Romulus and Remus arrived in October of 2024 and Khaleesi in January of 2025. They were about as close to actual living dire wolves as they could be. They live in a 2,000-acre Texas preserve with 10-foot zoo-grade fencing, cameras, drones, vets, care staff, a storm shelter. Colossal calls it functional de-extinction because they're not true dire wolves. They're wolves with more ancient-type traits designed to fill the same role in the wild. They're not Jurassic Park-level clones, but I mean, they're pretty damn close. Colossal Biosciences also gave us what they call the woolly mouse this year. Scientists compared DNA from around 121 mammoths and elephants, some fossils more than 1 million years old, to find out which genes made mammoths suited for cold. They picked 7 genes responsible for fur length, texture, color, fat storage, and then used CRISPR to edit them into regular lab mice. And the result was mice with long, wavy, golden fur and extra fat storage. In under a year, they had 38 of these woolly mice. Now, you can see where this is going. What we really want are mammoths. So why start with mice? Well, they reproduce quickly, about a 20-day pregnancy, so Colossal can test if the edits work in a live animal before trying them in larger species. The next step is Asian elephants, whose DNA matches mammoths 99.6%. They plan to edit 65 to 85 mammoth traits into elephant embryos, then implant them into pregnant elephants. Their goal is to have an actual mammoth calf by 2028. Maya was born on June 10th, 2022. She's the world's first cloned Arctic wolf. The team behind it was Syngene, a biotech company in Beijing. And they pulled it off using skin cells from a wild Arctic wolf that had died. They took that DNA and basically swapped the nucleus from the wolf cell into an egg cell that had its own DNA removed, and then they created the embryos. They implanted those embryos into beagle dogs, and it worked. Maya came out healthy. She's not de-extinct since Arctic wolves still exist in the wild, but this was a huge moment for cloning endangered animals. These wolves live in brutal, frozen environments, and their numbers are starting to shrink because of habitat loss and climate change. But Maya proves we've reached a point where cloning can realistically help endangered species from disappearing completely. The Tasmanian tiger, also called the Thylacine, looked kind of like a skinny wolf with stripes on its back, but it wasn't a wolf at all. It was a marsupial, basically a mammal that carries its babies in a pouch, like a kangaroo. These things used to roam Australia, but after years of hunting and habitat loss, the last known one died at Hobart Zoo in 1936. For a long time, that was the end of the story. But now scientists are trying to bring it back. In 2017, a group in Australia managed to sequence the Thylacine's DNA using bits from old museum specimens. Then they started editing the DNA of the Thylacine's closest living relatives, like the Tasmanian devil, to try and build a working embryo. They're using CRISPR to swap in Thylacine traits, hoping to eventually grow one either in a surrogate animal or maybe even an artificial womb. In 2021, a biotech company called Oxitec released genetically modified mosquitoes in the Florida Keys. The goal was to reduce the population of Aedes aegypti mosquitoes, which spread diseases like dengue and Zika. These lab-engineered male mosquitoes were designed to mate with wild females, passing on a gene that would prevent their offspring from surviving to adulthood. The idea was that over time the mosquito population would decline, but the plan didn't go exactly as planned. Some scientists were worried that the gene might not be as lethal as expected and that some offspring would survive and potentially create a hybrid population. And how would the reduction of one mosquito species affect predators or allow potentially more dangerous mosquito species to overpopulate? Even with these concerns though, the project moved forward and millions of genetically modified mosquitoes were released. It's only been a few years, so the long-term effects remain to be seen. In August of 1955, a woman named Naomi Johnson went swimming in the Ohio River near Evansville, Indiana, and came out with a story that's still being talked about today. She said something grabbed her from below, yanked her underwater, and tried to drag her down. She fought her way free and made it back to the surface, but her leg was covered in scratches and stained with this green slimy residue that wouldn't wash off for days. That alone would have made it a weird local mystery, but then it's just got stranger. Naomi later claimed a man in a military uniform came to her house, said he was with the US Air Force, and told her not to talk about what happened. No official report was filed and nobody could explain what she'd seen. Some people connect this to UFO sightings in the area around that time, saying it could have been part of a larger cover-up event. Others think that this was just a case of someone getting tangled up with a big fish or a log or seaweed. Still though, doesn't really explain the military visit part. Very weird story. From the late 1990s up until 2013, people in the Swiss village of Mal kept reporting a strange figure walking through the woods. They called it Le Loyon, or the Ghost of Mal. Witnesses described it as a tall person in a military-style cloak wearing a creepy old-school gas mask. They'd be completely silent, never speaking, just walking the same path over and over. No one knew who or what this was. Then in 2013, someone finally snapped a photo. A few weeks later, the sightings stopped. All that was found was a pile of the costume left in the woods, along with a note that basically said the person behind it was sick of being harassed and misunderstood. Some say it was just a harmless eccentric who didn't want to be bothered. Others think it was performance art or some type of social experiment, but there's another theory that something was going on in those woods. I mean, why walk around in a gas mask unless there's a very good reason? Was there some sort of experiment going on? It's really strange, because if it was just a hoax, I mean, just, some weirdo dressing up to scare people, you'd think they would have called it quits way sooner, but this lasted over a decade. There are some real accidents connected to the Pope-lick monster, but just what in the hell is the Pope-lick monster? It's a creature said to haunt a train trestle bridge in Louisville, Kentucky. People who've claimed to see it describe some kind of goat-man hybrid, has hooves, fur, horns, and the face of a man. The story goes that it lures people onto the tracks using hypnosis or by mimicking people, and then they get hit by an oncoming train. It sounds pretty out there, but people have actually died on that bridge. The train line is active, and because of the legend, people started going there to try and spot the beast for themselves. In 2016, a young woman was killed while investigating the site with her boyfriend. Another person died years earlier under similar circumstances, and some say the creature is just a spooky story to scare kids away from a dangerous area, but the origin of the legend is still murky. It goes back decades with no clear first report. So was it made up entirely? Or did someone really see something weird out there in the woods once and the story just grew from there? Whatever the truth is, the combination of actual deaths and the eerie setting has helped keep the Pope-lick monster legend alive. We've veered pretty far into the cryptozoology, urban legend realm, so let's talk about something a little more science-based. and down to earth, synthetic biology in general. This is basically when scientists build or redesign living things from the ground up. Might tweak bacteria to make medicine or design microbes that eat plastic. Sounds helpful, and sometimes it is, but the problem is once you mess with life at that level, you can't always predict what's going to happen. There have been real concerns that some of these lab-made organisms could escape into the environment and do things they weren't intended to do, like spreading out of control, mutating, and wiping out other species. One example people talk about is modified yeast strains used to make biofuels, so tweaking the yeast genes so that it produces stuff like ethanol or other chemicals that can be used for fuel. Pretty cool. Even scientists working in this field admit that we're still figuring out the risks. Most of the time, things go as planned, but if just one of these creations gets out and starts doing something unexpected, things can spiral fast. In November of 2024, chaos broke out at the Alpha Genesis Research Facility in Yamasee, South Carolina, when 43 rhesus macaque monkeys escaped after a caretaker reportedly left a door unlocked. The monkeys ran off into nearby wooded areas. 35 were rounded up pretty quickly, but 8 of them were on the loose for weeks. These macaques are very intelligent animals, and this was concerning because they can carry diseases transmissible to humans. So folks were pretty concerned as days turned into weeks. But what exactly were these monkeys being used for? Well, the facility does research involving viruses, bioweapons. So yeah, this was starting to sound like the beginning of Planet of the Apes. Meanwhile, animal rights groups were all over this, calling out the lab for being sloppy and putting the public at risk. And of course, they were already pissed about monkeys being tested on in the first place. In the end, all the monkeys were safely recaptured, but this was definitely not a good look. In early 2025, Colossal Biosciences made headlines after announcing that they had successfully brought back the direwolf, or something like it. Using advanced gene editing, they took gray wolf DNA and basically tweaked it until it resembled the long extinct direwolf. Bigger build, stronger jaws, different fur. Not exactly a clone, but close enough that it was a pretty big deal. The company called it de-extinction. Some scientists called it more of a Frankenstein DNA project, but either way, the animals are real. They're alive, and they're not planning to keep them in a lab forever. Colossal said the goal is to reintroduce these wolves into the wild at some point to restore balance to certain ecosystems, but that raised a ton of red flags. First off, no one really knows how they're going to behave in the real world. What happens if it mates with modern wolves, throws off the predator-prey balance? There's also the question of what happens if these things turn aggressive. Colossal insists that they got it under control, but when you're resurrecting an ancient predator in a lab, some people feel pretty iffy about it. Not me though, I think this is awesome. Between 1966 and 1967, people in Point Pleasant, West Virginia kept reporting something strange, a tall creature with glowing red eyes. It was humanoid, but it also had giant wings. They called it the Mothman. Some said it chased their cars, others claimed it flew over buildings or hovered near the old TNT plant outside of town. Dozens of reports came in over the course of about a year. Then in December 1967, the Silver Bridge suddenly collapsed during rush hour, killing 46 people. And this is when the Mothman legend really exploded, because some claimed that they'd seen the creature perched on the bridge just before the disaster. One of the biggest rumors surrounding the mysterious Mothman creature is that it was the result of a messed up government experiment. But whether it was a misidentified bird or something else entirely, nobody ever got a solid explanation. In April of 1973, a man named Henry McDaniel from Enfield, Illinois, called the police in a panic, saying that some bizarre creature had tried to break into his house. He said it had 3 legs, short arms, and glowing red eyes. He fired at it, but said it just jumped away unfazed. A few days later, some local kids said that they saw something similar near an abandoned house, and pretty soon word spread and people from outside the town started showing up, hoping to spot it. Some thought it might have been an escaped kangaroo, others leaned into the weirder theories, like it being a government experiment that got loose, or something not of this world at all. Interestingly, the area was near a military base, so that definitely helped with the government experiment angle. The government tried to downplay the whole thing, of course, but people who saw it stuck to their stories. Even the cops admitted that McDaniel didn't seem like someone making things up. To this day, nobody really knows what happened that that spring. In the summer of 1988, a teenager in Lee County, South Carolina, claimed that this huge creature attacked his car near Scape Ore Swamp. He said it was about 7 feet tall, covered in green, scaly skin, and had glowing red eyes. His car was damaged, there were these deep scratches all over it, and his side mirror was busted. Not to mention that the kid was clearly shaken up. Police wondered if he was just making up the story to cover up an accident, but this was a pretty lame excuse to come up with. I mean, a lizard monster attacking your car? They didn't pay it much attention, but after that, more people started coming forward with similar stories. One woman said something ripped the chrome off her car. Another man claimed he saw the thing staring at him from the edge of the swamp. Locals started calling this creature the Lizard Man. Police started looking into it now as more reports just kept rolling in. One of the sillier explanations at the time was that people were actually seeing a bear, which is a pretty lazy explanation. Again, there's an escaped lab experiment theory for this one as well. As fast as it started though, the sightings stopped. It's since become a local legend. As for what people were really seeing though, who knows? I can safely say it probably wasn't a bear though. The Restrador Basilero is a hunting dog that was bred in the 1950s to chase down jaguars and wild pigs in Brazil's thick forests. But by 1973, the poor dogs got hit with tick-borne diseases and a bunch of insecticide poisoning to try and get rid of the pests. Well, this was enough to wipe them out, and they were officially declared extinct. In the early 2000s, though, a group of dog lovers and breeders formed a group called the Brazilian Tracker Rescue Support Group. They set out to find any surviving dogs that had the original breed's traits in them. They spent years tracking down these dogs, selectively breeding them, and by 2013, they'd successfully brought the breed back from extinction. The Floriana giant tortoise lived on Floriana Island in the Galapagos. As the name suggests, these were huge tortoises, about 4.5 feet long. Sadly, by the 1800s, they were wiped out, mostly thanks to overhunting by sailors and the introduction of invasive species. And for over a century, everyone thought they were totally gone. But in 2012, scientists discovered that there might be some hope after all. They found a female tortoise on another island, Espanola, that had DNA showing she was related to the Floriana tortoises. This tortoise was nicknamed Lonesome George's cousin, and scientists went to work on a plan. They started breeding her with tortoises that were genetically close enough, and thanks to years of careful breeding, they managed to create a population of tortoises that closely resembled the extinct Floriana giant tortoises. It wasn't a perfect clone job, but was close enough to bring these species back in a new form. So while we don't have the extinct Floriana giant tortoise wandering around exactly, it's still better than nothing. Okay, now here's one you probably haven't heard of, the Montreal melon. So this is a type of melon that was native to Montreal, obviously, and used to grow in the area back in the 1800s. The melon had a very unique flavor. It's been described as having a slight spice to it, almost like nutmeg, and apparently it would just melt in your mouth when eating it. No surprise, it was a local favorite. But this melon pretty much disappeared in the early 1900s. No one really knows exactly why. Maybe it was due to disease or just the changing farming practices at the time, but whatever the case, the Montreal melon was gone. For over a century, it was nothing but a legend. But in 1996, seeds were discovered at a seed bank in, try and guess where, I'll give you a second. That's right, Iowa. Well, soon enough, the Montreal melon was back on local menus. The York groundsel was a little yellow flower that was hanging on in some pretty unusual places in York, England. It was a cross between two other plants and would grow in strange areas like railway sidings and roadways. But like many things in the world of nature, it didn't stand a chance once the city decided to crack down on weeds. By 1991, the flower was officially declared extinct in the wild. But just because it was gone doesn't mean people gave up on it. Turns out, there were seeds stored in the Millennium Seed Bank, a place where scientists keep seeds from all sorts of plants for situations just like this. In 2023, experts from Natural England decided to give the flower a second shot at life. Using those stored seeds, they managed to grow the York groundsel back to life, making this the first ever successful de-extinction in Britain. Now the plants are being reintroduced, and it's a major win for conservation, proving that with a little help from science, you can sometimes bring back what was thought to be lost forever. So sure, it's not an animal, but still a living thing, and that's pretty cool. The coelacanth is probably one of the most famous rediscovered animals in the world. For centuries, scientists thought that the coelacanth was long extinct, way back since the end of the Cretaceous period, around 66 million years ago. But in 1938, something that could only be described as mind-blowing happened. A fisherman off the coast of South Africa caught this unusual fish, and after scientists carefully examined it, they were amazed to find that this was a coelacanth. A fish that was supposed to have been gone for millions of years was still out there swimming in the ocean, and since then, scientists have tracked down several other coelacanths in the waters around Africa and Indonesia, proving that this ancient creature somehow survived the mass extinctions. So yeah, this one isn't technically de-extinct, just rediscovered. Still though, does make you wonder what other seemingly extinct animals may actually still be out there. So guess what? Scientists just brought back the direwolf. Yes, the actual creature. This is not something they're close to doing, they've actually done it. Colossal Biosciences, a Dallas-based biotech company, announced that they've successfully used ancient DNA to create 3 direwolf puppies. CEO Ben Lamb said, "Our team took DNA from a 13,000-year-old tooth and a 72,000-year-old skull and made healthy direwolf puppies." He also added, "It was once said any sufficiently advanced technology is indistinguishable from magic. Well, today our team gets to unveil some of the magic they are working on." So 2 of the pups are named Romulus and Remus. The third was named Khaleesi, of course a nod to Game of Thrones, which features direwolves. In fact, George R. R. Martin, the author of Game of Thrones, actually got to hold one of these pups. How nuts would that be? You write a fantasy novel with these long extinct creatures in them, and then decades later you actually get to hold a real living one. I don't think any goal I can imagine is ever going to live up to that. So these pups aren't clones. They're actually genetically engineered to be as close to direwolves as science can get. They were born from surrogate dogs and raised with bottle feedings, so they're friendly for now, but they're already starting to act more like true wolves. Thankfully, they live on a massive 2,000-acre preserve with cameras, drones, and guards keeping an eye on them. The Bucardo, or Pyrenean Ibex if you're feeling fancy, was a wild mountain goat that went extinct in 2000 when the last known female, named Celia, was found squished under a fallen tree in Spain. Brutal way to go, honestly, but science wasn't ready to let her go just yet. In 2003, researchers went Jurassic Park on the situation, and they took frozen skin samples from Celia, using them to create a clone. And it worked, sort of. A domestic goat was used as the surrogate mom, and against all odds, a cloned baby Ibex was born. It was the first time an extinct animal had been brought back to life. The victory party didn't last long though. The clone only lived for about 7 minutes. It had a severe lung defect and couldn't breathe properly, so technically the Pyrenean Ibex became the first animal to go extinct twice. Kind of amazing and kind of heartbreaking. Even though the success was short-lived though, it proved that de-extinction wasn't just science fiction anymore. It was messy and complicated, sure, but it was real, and a real possibility to do better. And in those 7 minutes, the seemingly impossible actually happened. So pronounce something like Przewalski. Przewalski's horse is a prehistoric-looking beast that once roamed the steppes of Central Asia. These stocky horses are the last truly wild horse species on Earth. Not wild as in used to be tame and then went rogue, wild as in never domesticated. But things didn't go well for them once humans showed up with fences and firearms. By the late 60s, these horses were completely extinct in the wild. The only ones left were in zoos, but in 2020, scientists stepped in. Using 40-year-old frozen cells from a stallion named Kaporovic, they created a clone, and boom, you had a healthy baby Przewalski's horse, now born in a Texas facility, who they named Kurt. Kurt's genes came from a bloodline that hadn't been part of the breeding pool in decades, so he brought much needed diversity to a population that was getting a little too inbred. And thanks to years of conservation work, these wild horses are back in their native Mongolia, running free once again. The quagga was a strange-looking animal, a zebra subspecies with stripes in the front half of its body and then just a plain brown rear end. It once lived in the grasslands of South Africa, roaming around in large herds, but like a bunch of species, it didn't survive European colonization. Settlers hunted it for meat and hides, and by the 1880s, it was declared extinct. The last known quagga died in captivity in Amsterdam in 1883. For a long time, scientists didn't even realize the quagga was a distinct subspecies. That changed in the 1980s though, when a German natural historian named Reinhold Rau launched the Quagga Project. Using preserved skins and taxidermy specimens, Rau conducted genetic testing and confirmed that the quagga was genetically close enough to plain zebras that selective breeding might be able to bring them back, at least in appearance. The idea was to breed plain zebras that naturally showed fewer stripes, generation after generation, until the animals started to resemble the extinct quagga. Over the decades, the project produced zebras with lighter coats, reduced striping, and even that same body shape described in old quagga records. The animals born from this project are now called Rau quaggas, and sure, they aren't true quaggas in the genetic sense, but they're close. Some have even been reintroduced into the environment in South Africa, living in the same type of environment their ancestors did. The Lord Howe Island stick insect was once believed to be extinct, wiped out by invasive rats and other pests. The insect, also known as the tree lobster for its very weird lobster-looking appearance, lived on Lord Howe Island in the Pacific Ocean. By the 1920s, the insect had completely disappeared though, or so everyone thought. For decades, it was assumed that the rats had done them in. But then in 2001, there was this incredible discovery. Researchers came across a population of these thought to be extinct insects surviving on Ball's Pyramid, a jagged, steep rock formation off the coast of Lord Howe Island. The fact that these things managed to hang on in such a harsh, isolated environment was mind-blowing, as well as the fact that they were even there at all. So now, scientists have been working hard to breed them in captivity and reintroduce them to their original island home, and they're slowly making a comeback.
What if fish evolved to walk on land? Sounds like something out of a movie, but it is entirely possible, and there are people out there who actually believe that this already happened. Fish are already known to use primitive forms of walking, like mud skippers who use their fins to move across land. Don't get me started on the smooth handfish either. In fact, certain species have evolved to do this very thing, and if humans were gone, evolution might push this characteristic even further. Over time, fish might develop stronger, more efficient limbs to handle terrestrial environments. These walking fish could live in shallow waters and on land, adapting to ecosystems where air breathing becomes as necessary as swimming. Some scientists believe that fish could evolve into amphibious creatures similar to early vertebrates that first left the oceans. Walking fish could become one of the new apex species, filling the roles that mammals and reptiles once held. Picture a future where fish leave the water completely, evolving into creatures with complex land-based behaviors. They might even develop the ability to climb, interact socially, and compete for resources like we see in mammals today. Imagine enormous frogs, salamanders, and newts roaming the planet with sizes so intimidating they could rival the great dinosaurs. After humans disappear, scientists think amphibians might grow into giant versions of their ancestors. The carbon-rich atmosphere after the extinction of humans could result in larger body sizes, like the gigantic amphibians that once roamed the Earth during the Carboniferous period. These massive creatures could dominate freshwater ecosystems, becoming the apex predators of their environment, with abilities to thrive in both water and on land. What if the ocean's most intelligent creature became even smarter? We kind of touched on this earlier, but octopuses are already considered one of the most intelligent marine animals with remarkable problem-solving abilities and complex behaviors. With humans gone, these remarkable creatures might not only survive, they could thrive. Octopuses could even evolve further, developing higher cognitive abilities that could enable them to manipulate their environments in ways that we haven't even yet imagined. They could use tools to build shelters, make weapons, or even engineer underwater cities. These creatures already display behaviors like opening jars, camouflaging themselves, and evading predators with precision. I mean, have you seen my octopus teacher? In a world free of human interference, octopuses could become the dominant species of the oceans. Their intelligence, coupled with their adaptability, might even lead to social structures where groups of octopuses collaborate to achieve goals and pass down knowledge, creating a whole underwater civilization. Would you live in the octopus world if you had the option? Let me know down below in the comments. I think I would. I think that would be a peaceful world. Because they're very smart, you know what I mean? Anyways, weird tangent to go off on. What if giant predatory birds make a comeback and rule the earth just like ancient times? Imagine giant birds, like the massive terror birds from the Cenozoic era, returning to Earth. With the extinction of apex predators, birds could evolve to fill those ecological niches once dominated by mammals and reptiles. These birds could grow massive, some species potentially reaching size comparable to modern-day ostriches or emus, with sharp beaks and talons capable of hunting large prey. Could sharks evolve to live on land? It might sound absurd, but the future is full of possibilities. Sharks are already incredibly successful apex predators in the ocean, but what if they adapted to life on land? In a world without humans, the evolutionary pressures could push sharks into new terrestrial environments, potentially adapting to land-based life. Over time, sharks might evolve modified fins that function as limbs, helping them traverse the land in search of food. Imagine a massive land-dwelling shark with a body built for both water and dry environments. This is giving Tralalero Tralala. If you know, you know. These land-based sharks could hunt large terrestrial prey, using their incredible speed and sharp senses to track down and overpower animals on land. Just as the first fish began their journey to land millions of years ago, these sharks might be the next step in evolutionary history. Their remarkable ability to adapt, combined with the right environmental conditions, could lead to them becoming the dominant predators in a post-human world. We've all heard the phrase, the insect apocalypse, but what if insects grew to monstrous sizes? In a post-human world with oxygen levels in the atmosphere increasing and a lack of predators, scientists believe insects could grow to enormous sizes, much like they did during the Carboniferous period. These giant insects could dominate ecosystems, with some growing to the size of large birds or mammals. Imagine ants the size of dogs or dragonflies with wingspans the size of a human's arm.
After reviving dire wolves, Dallas-based Colossal Biosciences is going even bigger. They've now set their sights on two massive Ice Age animals, the short-faced bear, which stood about 14 feet tall and weighed over a ton, and Castorides, a beaver that was the size of a small person, meaning like 5 feet tall. Their plan is to basically take ancient DNA from fossils, find the genes that control things like size, metabolism, and bone structure, then plug those into modern animals. Grizzlies stand in for the bear, regular beavers for the mega beaver. They use CRISPR to do the editing, implant the embryos into surrogates, and hope the result looks and acts like the original Ice Age version. The idea is that these animals had very positive effects on the environment. They shaped rivers, moved seeds around, and cleared space for other species. Colossal's even working with indigenous groups to figure out how and where these creatures might fit back into the wild. Still, there are big unknowns. These animals disappeared over 10,000 years ago, so drop them into today's forests or waterways, and honestly, who knows what happens? They could throw things off completely at worst, or just not survive. Now, over in Australia, scientists have been trying to bring back one of the weirdest frogs I've ever heard of, and I've talked about some weird frogs on the channel before. Now, it's called the southern gastric brooding frog. It went extinct in the early 80s, but what was so strange about it? Well, it swallowed its own eggs, shut down its stomach acid, and grew the babies inside of it. Then it would give birth by burping up its young. That has to be one of the craziest birthing processes I've ever heard of. Since 2013, a team working on the Lazarus Project has been trying to bring it back using preserved tissue samples from the 70s. They took the nuclei from old cells and put them into eggs from a closely related species, the barred frog. Now, a few embryos started developing. They reached an important stage called gastrulation, where the cells finally start folding into organs. Unfortunately, none made it to full tadpole tadpoles, but even getting that far was a huge deal. It proved that it's possible frozen cells from an extinct species can still form life. Now, according to project lead Dr. Mike Archer, the science isn't being held back by DNA damage, it's the technology. Frogs only lay eggs once a year, so there's a really small window to get it right. For now, they're kind of stuck with embryos that stop halfway, but even that's enough to show amphibian, resurrection. It isn't completely unheard of. Now, if they can get past the early development stages, the next steps are growing live tadpoles and eventually froglets that can make their way back into streams. In April of 2025, Colossal Biosciences announced that they'd cloned 4 red wolves using so-called ghost alleys found in coyote wolf hybrids along the Gulf Coast. Red wolves are critically endangered. There's less than about 20 left in the wild and just a few hundred in captivity, and their gene pool is super shallow. What exactly are ghost allies though? Well, they're basically hidden bits of red wolf DNA that can still be found in coyotes, so Colossal extracted them, then used domestic dog surrogates to bring 4 new pups into the world. Now, these pups now live on the same Texas preserve than their dire wolf clones Colossals they created. Conservationists call this a form of genetic rescue, so using a natural genetic variation to boost the health of a dying species. Critics say we still don't know how these clones will do in the wild, but everyone agrees this is a major step forward when it comes to conservation. Now, the Javan Batang is a type of wild cattle native to Southeast Asia. Their numbers have been dropping. Back in 2003, they were brought back thanks to cloning. Scientists used skin cells from a Batang that had died in 1980 and preserved in the San Diego Zoo's Forest Frozen Zoo. Now, they transferred the nucleus from that skin cell into a domestic cow egg, implanted it into a pregnant cow's, and voila! A live Batang calf was born on April 1st, 2003. A second calf followed 2 days later. Sadly, that one didn't make it. Now, the first calf did pretty well though. It lived a full 7 years before dying from a leg injury in 2010. So, while not perfect, I mean, these early days of cloning were full of failures. It wasn't a short-lived lab experiment either. This was a healthy, breathing animal that helped preserve, genetics that might have been lost forever. You could say it was sort of proof of concept. Now, scientists in Kashmir cloned a Pashmina goat named Nori to help protect their famous Kashmir goats. These goats produce that super soft wool people pay really big money for, but these goats are slowly dying out because of habitat loss and climate problems. Now, the cloning process involved taking a skin cell from a healthy goat, putting its nucleus into an egg cell, and then implanting that into a surrogate goat. Nori was born healthy and is growing like any normal goat, still producing that prized cashmere. If this works long term, the same method could help protect other endangered species like the Kashmir stag. I think the idea of cloning is often thought of as needless tampering just for the sake of it, but cases like this show it can really be a good thing. It can help keep animals from going extinct while also supporting the people who rely on them to survive.
In 2009, researchers in Russia and Belarus created genetically modified goats that could produce human breast milk. It wasn't exactly pure human milk, but it was close. It contained 60% of the proteins that make up human milk. Not long after, a team in China took things even further and created a whole herd of 300 cows that could also produce human milk. The idea was to be able to harvest human milk on a large scale, mass produce it, and then sell it in stores. I mean, they figured people would not only want to buy human milk, but human milk that came out of a genetically modified farm animal. The Chinese team originally planned to have their milk in stores by 2014, but the idea didn't exactly catch on as they'd hoped, and the plan got delayed. Back in 1996, a group of scientists at Harvard, led by Joseph and Charles Vacanti, created what became known as the Vacanti mouse. This mouse had a human-shaped ear growing right on its back. Now, this wasn't some weird mutation, it was a planned experiment. They took cow cartilage cells, placed them into a mold shaped like an ear, and then implanted it under the mouse's skin. The ear-shaped mold helped guide the cartilage into the desired shape, and as time passed, the cartilage naturally grew into that shape. The goal was to see if they could create artificial ears on mice and then graft them onto humans. The mouse used was what's called a nude mouse, an immunocompromised species that doesn't reject foreign tissue, so it was perfect for this kind of work. In an interview with Newsweek, Joseph Vacanti joked the mouse had the ear removed and then lived out a happy, normal life, which wasn't true. Lab animals are typically euthanized after experiments, and the original paper mentioned the mice after sacrifice, a term used to describe when animals are killed post-experiment. Ilya Ivanov was a Soviet biologist in the early 20th century who became infamous for his attempt to create a human-ape hybrid. Back in 1910, Ivanov gave a talk about the possibility of creating a human-ape hybrid by using artificial insemination. Then, in the 1920s, he took his theories to French Guiana, where he carried out a series of experiments. He tried inseminating female chimpanzees with human genetic material, but this was unsuccessful. After that, he went back to the Soviet Union and started trying to organize similar experiments, only this time he'd flipped them around, injecting ape genetic material into human volunteers. These plans were thankfully interrupted when the last of his orangutans died. Ivanov's work wasn't just controversial, it was downright disturbing to many, and it led to his eventual arrest and exile. Just this year, scientists at the University of Tokyo managed to combine chloroplasts, which are green energy-producing cells found in plants, with animal cells. The idea here was to give animal cells the ability to create energy from sunlight, a process called photosynthesis, just like plants do. For years, scientists had tried to mix chloroplasts with animal cells, but the animal cells would just digest the chloroplasts within a few hours. That was until the Tokyo team used a specific type of algae called this thing here. But this specific algae could stay active in animal cells. They inserted these chloroplasts into Chinese hamster cells, and to their surprise, the chloroplasts didn't just survive, they kept performing photosynthesis for up to 2 days. The hamster cells even grew faster than usual. It looked like the chloroplasts were giving them some extra energy. Researchers still aren't sure exactly how the chloroplasts and the hamster's mitochondria interact, but they're working on it. And these plantable cells could possibly help us find new ways to create tissues or even new forms of energy. In 2019, reports came out about a team of researchers from the Salk Institute in the United States who'd successfully created the first human-monkey chimera. Led by Professor Juan Carlos Psua Belmont, the team had already produced embryos with small amounts of human cells in pigs and sheep, but this time they went a step further, working with monkeys. The idea was similar to their previous work, to create chimeras that might one day be used to grow human organs that could be used for life-saving transplants. These human-monkey chimeras were reportedly only allowed to develop for a few weeks though. The process was carefully monitored and stopped before any of the embryos developed a nervous system or organs, which could raise more serious ethical concerns. These chimeras were also created in China to avoid legal restrictions in other countries. The difference in evolutionary distance between humans and monkeys is much wider than between humans and chimpanzees, but it still opens up the possibility of growing organs for transplants. The chances are slim though that human-monkey hybrids could be brought to full term because of the vast amounts of genetic differences between the species.
Starting us off at number 10, we have the quagga. Once a prolific subspecies of zebra across South Africa, the quagga was sadly hunted to extinction in the 19th century. The decline in population all started after European colonists took to South Africa and hunted the species nearly into the ground as it competed with domesticated animals for forage. By 1878, the wild population was decimated, although some were taken to zoos in Europe in an effort to rehabilitate, but tragically in the end, the breeding programs were unsuccessful and the last captive quagga died in Amsterdam in 1883. That was until about 100 years later when the quagga became the first extinct animal whose DNA was analyzed, and it was then that scientists discovered just how close a match they were to the plains zebra. By 1987, the Quagga Project began, and their mission is to recreate the phenotype of hair coat pattern by selectively breeding the genetically closest subspecies, which, as we now know, is the zebra. The first foal of this project was born in 1988, and since its early days, they have seen quite a bit of success. If all goes well, the selectively bred quagga, like zebras, will be released back into the wild. Coming in at number 9, the Cuban macaw. Prior to the 15th century, these vibrant and colorful birds could be found all over the main island of Cuba. But when Europeans landed, they had other plans for these parrots. Although indigenous populations had hunted the species prior to the colonists' invasion, once the Europeans arrived, the amount soared. Many were hunted or traded, while others were sent back off to Europe to live as cage birds, and eventually, by the late 19th century, between the rampant overhunting and habitat destruction, the very last of the Cuban macaws died, making the species extinct. But according to the British writer Errol Fuller, aviculturists are rumored to have bred birds similar in appearance to the Cuban macaw using the genes of a sister species. So who knows, we could be seeing them fly around Cuba in the near future. Coming in at number 8, the Arabian Aurochs. Historically speaking, the Arabian Aurochs could have been seen roaming around throughout most of the Middle East. But by the time the late 19th century rolled around, you were not likely to see one outside of Saudi Arabia, and in the 1930s, the only remaining populations were found either in the Negev Desert in the north or the Rub al-Khali in the south. Tragically, it only got worse from there. Starting in the 1930s, it became fairly customary for Arabian princes and oil companies to hunt down the animal, and eventually the hunts grew to employ as many as 300 vehicles at once. By the middle of the 20th century, the northern population was effectively extinct, and finally the last Arabian Aurochs died in 1972. Thankfully, a few people had the forethought to send a couple off into captivity, and they were effectively reintroduced into the wild a few years later. That being said, they are still an endangered animal, but interestingly, it is the first animal to revert to a vulnerable species after being declared extinct. So it's definitely getting better. Coming in at number 7, Caspian tigers. During their prime, Caspian tigers could be found in Turkey and through much of Central Asia, including Iran, Iraq, and northwestern China. But with the Russian colonization of Turkestan during the late 19th century, their population began to be threatened. The first problem was obviously that the tigers were being hunted by large parties of sportsmen or military personnel. Until the early 20th century, the army was used to clear predators from forests around settlements and potential agricultural lands. In fact, until World War I, about 50 tigers were killed in the forests each year. By the 50s, they became an officially protected species, but even so, by the 1970s, the last remaining Caspian tigers were gone. However, according to recent findings that the Siberian tiger is the closest relative to the Caspian tiger, discussions have been started about introducing the Siberian tiger into a safe space in Central Asia. And hopefully, if they get it right, the tiger would adapt and live successfully where the Caspian tiger once roamed. Coming in at number 6, heath hens. Once an extremely common bird, heath hens were a subspecies of prairie chicken that could have been found just about anywhere across North America in colonial times. However, like many other species, the population was deeply affected by colonizers who hunted them extensively for food. In fact, it has been speculated that at the first Thanksgiving, it was actually heath hens that were served, not wild turkey. However, by the time the 18th century rolled around, the heath hens developed a reputation as being the poor man's food, as it was cheap and plentiful. And as the hunting continued to soar, the heath hens became more and more of a distant memory. By the 1870s, the animal was virtually extinct from the mainland, with only a few hundred left on Martha's Vineyard, until eventually the late 19th century put a hunting ban in to try and keep the species alive after an estimated 70 remained. This worked for a while, but due to a myriad of problems, the last remaining heath hen died in 1932. But there is still hope for them to return. As they are closely related to the prairie chicken, scientists have started researching projects aiming at the de-extinction of the animal using DNA from preserved cells as a basis for restructuring the DNA of greater prairie chickens. So with any luck, heath hens could be roaming around soon again.
Now, China's been cloning animals way longer than most people realize. All the way back in 1963, they successfully cloned a common carp using nuclear transfer, a huge deal at the time. Now, cloning fish is trickier than mammals because of how they reproduce, but they pulled it off, and it laid the groundwork for a lot of what came after. Since then, they've cloned other species too, like Mozambique, tilapia, brown rats, and even pigs. They were mainly trying to make fish farming more efficient and get a better handle on how these animals reproduce. By cloning fish, scientists could create more consistent breeding stock, which matters when you're, feeding just millions of people, no big deal. It also opened the door for conserving endangered fish species later on, so while cloning tilapia didn't get the attention that, say, cloning a mammoth might, it was quite a big step. The Florina giant tortoise used to live in Florina Island in the Galapagos, but it was wiped out back in the 1800s, mostly thanks to whalers and pirates who used them as food and introduced invasive animals. But now scientists are bringing it back, not through cloning technology, but through smart breeding. In the early 2000s, researchers found tortoises on Isabella Island, about 50 miles away, with Florina DNA. Turns out sailors used to move tortoises between islands or just dump them off when they didn't need them anymore. These tortoises weren't 100% Florina, but some were pretty close. So scientists gathered, the ones with the strongest genetic similarities and started a breeding program on Santa Cruz Island. As of 2025, they've got about 400 baby tortoises with high percentages of this Florina DNA. Now the plan is to release them back onto the island, but first they need to get rid of rats, cats, and other invasive animals that would basically just eat the babies. This matters because these tortoises are what scientists call ecosystem engineers. They stomp around, spread seeds, clear brush, and basically keep the environment in check. If all goes well, this will be one of the most successful island rewilding projects ever. Way back in 1952, two scientists named Robert Briggs and Thomas King pulled off something huge. They cloned a frog using a method called somatic nuclear transfer. Basically, they took the nucleus, the cell's control center, from a normal frog cell and put it into an egg cell that had its own nucleus removed. That egg then started developing into a frog embryo. It wasn't a full animal clone, but it showed for the first time that you could take a mature cell, reset it, and grow a new organism from it. This was a massive breakthrough because it laid the groundwork for cloning animals later on. It proved that cloning could actually work. Plus, frogs are simpler to work with in the lab than mammals, so this gave scientists a practical way to explore cloning. And then about a decade after the first frog cloning, scientist John Gurdon came along. Instead of using embryonic cells like before, he took a fully specialized adult cell from a frog's intestine and transplanted its nucleus into an egg cell. The egg grew into a tadpole, proving that adult cells, even after they've kind of decided what, they're supposed to be doing, and so to speak, still hold all the information in their DNA to create a whole new animal. Before this, most scientists thought once a cell, specialized, it lost that ability. Now, Gurdon's experiment showed cells could be reprogrammed, which was huge for cloning. This discovery directly paved the way for cloning mammals, including Dolly the sheep, decades later. Now, without this experiment, cloning adult animals probably wouldn't have even been possible. In 2025, scientists put together a big review looking at cloning and conservation. They found that so far, 56 different species have been cloned using nuclear transfer techniques. Now, that's everything from mammals and amphibians to fish and insects, and about 90% of those cloned animals lived full, healthy lives. Plus, most of them ended up being able to have babies of their own. Love that for them. That's huge. Cloning is actually making a difference in real-world conservation. Scientists are using cloning to bring back genetic diversity in endangered species, revive species that are struggling, and we're on the brink of actually bringing back animals that went extinct a very long time ago. Cloning gives conservationists another tool in their toolbox, especially for species with tiny populations or ones that can't breed naturally anymore. The review also points out that the technology is getting more reliable and accessible, so cloning will probably play a bigger role going forward.